Ladies and gentlemen, welcome to the second semi-final of FameLab International 2014. Now please put your hands together to greet your host for this evening, Quentin Cooper.
Good evening, good evening, good evening. I have just come here straight from the event that was here previously, Sean Carroll's Guide to the Higgs Boson. Was anybody in that one? Yeah, is your mind still slightly blown? Yes, so mine is gradually recovering. So we all share a field. The field has been dealt with, so we can take that as granted. So who was here last night for the first semi-final? Woo! Excellent. We like this. We might get recommissioned for a new series if this was HBO. And who has wandered in here for the first time and is new to the whole competitive science communication thing? Oh, that means I've got to explain the rules again. What a pity. I'm always hoping to skip this bit. Okay, well, despite this being in the Cheltenham Science Festival programme as being FameLab Semi-Final Part 2, it's not Part 2. It's a brand new semi-final. It's not like we're going to bring out all the people from last night again. This is a new one as well. All new FameLab champions. We've got 12 of them representing, slightly confusingly, 14 countries. Each has won through local heats and national finals for the right to be here. But as that prefix semi in semi-final implies, only half of them, in fact less than half of them, 5 of them are going through to Thursday's final. This is the event that decides it. Now tonight, as they did in all those heats and regional finals, what they have is 3 minutes to impress and amaze yourselves and our judges by getting across in an engaging, entertaining and entirely accurate way some aspect of science, technology and or maths in at most 3 minutes with no props other than what they can carry on, no PowerPoint, no pets, no radioactive materials. If you go over 3 minutes, you hear this. If you go more than 15 seconds over 3 minutes, you'll see the 2 laser cannons positioned here. And apologies again to the relatives of the Slovenian semi-finalists from last night. That was just an accident. Now, there's probably a bit more to it than that, but you'll pick it up as you go along. Essentially, what we're trying to do is to go from 12 winners to 5 in a quick, serious, but slightly unusual way. It's a slim and earnest off-balance elimination, which spookily is an anagram of FameLab International second semi-final. You can work it out if you have time, but you won't. It's ultimately our 3 judges, though, who do that eliminating, but they will, of course, be influenced by your yelling, your clapping, your cheering, and to a lesser extent, your coughing and yodeling. So feel free to try all of the above as we bring on our obdurate triumvirate. We're going to start with our chair. She's the founding director of Science Made Simple, which has taken exciting hands-on science experiences to tens, if not hundreds of thousands of people. She's the winner of many awards, including the prestigious EU Descartes Prize for Innovative Action in Science Communication, and she's a multiple books author. Please welcome Wendy Sadler. Yes, we've actually turned your entry onto the stage into some sort of bizarre exercise here as you find your way on. Next, move one more round, if you would, Wendy, thank you. Next, the Professor of Biomedical Imaging at UCL, and according to his Twitter homepage, still co-director of this very science festival, Mark Lithgow. Notes to Mark updates your Twitter page. And a clue to the identity of our final judge is that this afternoon he tweeted FameLab UK saying, rushing back from Queen's speech to be there. Many locally and beyond will know him. Some locally but not beyond may even have voted for him. The MP for Cheltenham for the last 9 years, Martin Horwood. So Wendy, comparing people from different countries, different cultures, different levels of English skills, many performing in their second or third language. It's not a competition about who speaks the best English, though, so what will you be looking out for?
Well, I have to say I'm quite a sucker for a good prop, so I might be looking for some good use of props.
Oh, that's bad news for all those of you who haven't bought props. Right,
That's
okay.
not essential, that's not essential. And also just just charisma, really. Something where I just wanted to listen more to them. So 3 minutes isn't very much. If at the end I'd like to listen to them for half an hour, that's a good thing.
Right, so charisma with a prop, ideally. Mark, any other criteria you're looking out for?
Oh, I want to make me laugh. Make me laugh so my belly hurts so much. And, and I have a love of musicals. If any, I love musicals. And, you know, West Side Story was, Carousel and, you know, Oklahoma.
Right.
But so if you can sing, dance, I'm swooning, you know, they'd be great. Okay. Musicals.
Okay, so far we've got it's helium, it's helium, you know, it's okay, fine. Martin, now what do you bring? What's your perspective? Is this a key insight into the democratic process that you're going to be bringing us here?
No, I think I'm looking for an insight into the scientific process, and I want people who can communicate to a politician and a policymaker, which is roughly like communicating to a kind of 6-year-old, but without the good behaviour. You know, I think you've just got to make it really clear.
Okay, so clear, so it's clarity. We've got charisma, and we've got Hollywood musicals. I think the official third one is content, but Mark might turn it into Hollywood musicals, and a bit about props. Sorry if you haven't got any. It's a tough job, but these were the only people wonderful/gullible enough to do it. So please, some applause for our apocalyptic triptych, Martin Horwood, Mark Lithgow, Wendy Sadler. Right, let's get going with our first semi-finalists. Being here is the stuff that dreams are made of, which is the opening line of which classic movie? Am I the only classic? Sorry, it's the closing line. Humphrey Bogart film. Humphrey Bogart film, the stuff that dreams are made of? The Maltese Falcon. Thank you very much indeed. Which, of course, comes to my mind because our first dreamer, hoping to stuff everyone else, is our Maltese FameLab one. Now, besides being an anagram of moral parallel elitist, Maria Stella Portelli is a chemistry and biology graduate from Malta University, and she loves to travel. And when I say loves, I mean loves. She says it borders on addiction, although borders may just encourage her to go even further and cross them. She spent time at the University of Antwerp. She's taught English and science to children in India and worked in the flavour and fragrance factory in Poland, which is a hell of a tongue twister in the first line. She's now about to spend time in the even tastier and sweeter smelling and slightly noisier Winton Crucible in Cheltenham, but of course not too much time, just 3 minutes for our first FameLab semi-finalist of the evening, Maria Stella Portelli.
I must say I feel a bit like a party pooper, opening this evening with a topic as gloomy as hatred. But the truth is, hatred is all around us. All you have to do is open the newspaper. Actually, you don't even have to go that far. Just look around. And by this, no, I don't mean Cheltenham. Cheltenham is awesome, but think about the countless years of wars, the unspeakable acts of evil and atrocities. It makes me think that humans aren't so humane anymore, if that makes any sense. So why has this feeling of anger mixed with fear evolved? And I'm using the word evolved quite deliberately here, because it appears that indeed hatred is an evolutionary adaptation, one that evolved in hunter-gatherer tribes. So you see, in these primitive times, when a stranger walked into your territory, you wouldn't exchange a "hey, what's up?" or offer tea and biscuits. You would act like what we would nowadays call a total weirdo. But you would do this because you have a lot at stake. You have your food resources, you have your mate, and you have your territory to protect. So if you are not able to distinguish your friends from your enemies will outcompete you and will be the cause of your doom. So eventually, this feeling of distrust became instinctual, and an imprinted fear of outsiders developed. And the key to this is evolutionary fitness. And by fitness, I don't mean how much your gym membership is paying off. I think most of us have given up on that by now. But I mean the ability to survive and reproduce, and hence transfer your genes, the successful genes, from one generation to the next. So let's put it this way. If you have no problems with strangers walking in and out of your territory, you'd probably get yourself killed pretty quickly without leaving many offsprings behind. And if you attack neighboring communities and tribes, you'd get more territory, and hence you'd get more food resources, and potentially also more mates. So you would be increasing your evolutionary fitness. So If there is a genetic basis for traits like aggressiveness and fearfulness, these would be promoted, and eventually these would become the norm. On the other hand, and this is my attempt to not sound so gloomy, just like hatred, distrust, and violence evolves between different tribes, love and trust evolves within the members of these tribes for the same reason, for the survival of the tribe. So my final message is this. Even if hatred is a product of evolution, evolution itself is a product of constant change and adaptation. Hence, as we have evolved to hate, we can adapt to not hate. After all, we can see ourselves as just one big tribe. So let's spread the love and stop this madness. Thank you.
Thank you. That was a great start. Hard to go first, so well done. And a nice evocative subject, I think, hatred. Not depressing, quite good. I'm interested then, so we've looked back at why this might have evolved. I'm not a geneticist, I'm not an expert at all. Where do we think it might go in the future? Will the way that we live now, will it affect how we evolve in the future?
Well, now we live in a society that has many rules and laws. So now many political issues come into place. And so when somebody does something which is against the law, we put him in prison. But if you think about it, it's our nature to be violent at some point. So that puts issues of whether putting people in prison is the right way to go. So this opens a lot of issues and a lot of discussions. And what the right answer is, I don't know. It's a really tough subject to discuss.
Okay, so on the subjects of hatred, trust, and survival, I think you're going to have to ask Martin and look to the politicians. But animals, I love animals, and I was trying to think: do all animals have this trait as well? Because I was looking at a load of ants the other day, and they seem a pretty cooperative bunch. They don't seem to have that trait.
But
do
like,
animals have it?
Some animals do, like chimpanzees. You see this in chimpanzees very well, very clearly. I mean, some studies are done on chimpanzees. But then when you think about it, like, if animals actually hate, like, have this spiteful feeling, I mean, I don't really know. I mean, I'm not the animal, but they do acts of violence on each other to get food, to get mates and everything. So I think it's quite a controversial subject at that.
I mean, I work in quite a tribal place. We do hatred quite well at times. Prime Minister's Question Time, very hate-filled. But actually, I mean, a lot of our best work is done when we're actually collaborating and cooperating behind the scenes. So, and that seems to be quite a trait in human society, doesn't it? So where's the evidence that this hatred is a natural part of evolution and not a malfunction caused by the fact that we now live in cities and drive cars and do all sorts of unnatural things like drink purple slushies and all that kind of stuff?
Because when there were tribes before, like, people would form groups. They would form their hunting tribes to go and get food and whatever. So they would be in competition with the other tribes. So that's how there is this feeling of spitefulness between them.
What's the evidence for that?
There is a lot of study between, like, other animals, like chimpanzees and things like that. And you can see that it's quite clear.
Okay, Martin, this is not a select committee, it's FameLab. I'll have to stop you there. Have to end this loving. Hate, hatred, love, Maria Stella Portelli. Maria, of course, from Malta, a small population completely surrounded by water. Unlike, oh, hang on. You hope you're all going to row home tonight. Now, having started a long way south and east of here in Malta, we're going to continue on the same bearing for about another 1,000 miles or so to Egypt. Many things have changed in Egypt in the last few years, but somehow FameLab has survived and thrived. And our 2014 winner is Ahmed Barra. Ahmed is studying medicine at Ain Shams University in Cairo, and he says he finds himself constantly rushing to friends to explain scientific ideas and principles and facts, and nothing satisfies him like hearing people saying, "Oh man, that's awesome," or "I understand now." So if everyone in the audience can decide which of those you're going to go with, "Oh man, that's awesome," or "I understand now," and shout it out after you've had 3 minutes in the company of FameLab Egypt champion Ahmed Barra.
Ladies and gentlemen, today we'll discuss the greatest mystery of our human body. Of course, it's the brain, the orchestra in our head. where all different nerve cells called neurons are like different musical instruments. Each make a different sound, and they all come together to form the music in our head in the form of electricity. And these patterns of electricity decide whether we're happy, sad, or depressed, or whether we're suffering from devastating diseases such as schizophrenia and autism. And all what we needed to know to solve this mystery is the function of one neuron, of each type of neuron, the sound of each musical instrument in this orchestra. And in a trial to do this, we inserted an electrode inside the brain and tried to excite only one type of cell, but it was unspecific at all, and it was a mess. And the answer was a beam of light. Lately, in 2005, researchers in MIT discovered that an algae was moving towards the light. And upon studying it, they found that they have a specific protein that activates the cell when a light was casted upon them. And they had a brilliant idea to take the gene, which is the recipe for this protein, and to insert it into a certain type of neurons into a rat's brain through a virus that acts like a messenger. And once this protein was expressed, and they inserted a source of light called an optic fiber inside a rat's brain, and no, I'm not a rat. So they shine a light, and something amazing happened. The rat would go into a rotatory movement. But what is the cause of that? It's caused by that a specific type of neuron, which is the cause of this rotatory movement, was excited. And finally, we could hear the sound of one musical instrument and know how it affects our behavior, memory, and learning. Furthermore, we can cure some diseases with this technique called optogenetics. We can, for example, if you know the Parkinson's disease, where patients have static tremors, we can insert this protein inside the neurons responsible, which are not working. For example, if one instrument was not working properly in this amazing orchestra, we will insert this protein inside these types of neurons and shine a light upon them, exciting them. And once they are excited, this will completely cure the disease. Optogenetics has radically changed the way we study the brain and how it functions. And the question remains, after applying this technique to humans, could we really control the brain? Could we really be the maestro of this amazing orchestra with a beam of light? Thank you. Thank you.
Oh, that was a great description of optogenetics, and I'm going to declare my interest in this because half of my lab does optogenetics, so you are going to get a slightly technical question now. One of the big problems with it, and it is beautiful, and you're right. You can literally put the animal on the bench, shine a light on its head, and it will run round in circles. And you can control the brain via light. The problem is that light doesn't travel very far in tissues. So if you are going to activate the brain, how do you get the light into the brain? If it's going to be used as a therapy, how do you really use this as a therapy?
Well, it's about, I didn't quite study well the way we can insert an optic fiber inside the brain, but it's about the part of the brain that you are trying to excite or work on. So it's about the delivery of the optic fiber and the frequency, its wavelength. It's important because with slight change in this protein channel rhodopsin, you can, instead of exciting the cell by passing inside the positive ions, you can inhibit the cell by passing in negative ions. So I think that the frequency and the intensity of the wavelength going from the optic fiber is really important, and the place of its delivery, like I don't know how it's said in English. It's the place where we can deliver the light. So that's what I think.
Have you just read about this, or do you actually do work on it?
I've actually read about this and did a lot of research about it, and thought it was it was the method of the year for Nature magazine. Yeah, so I really liked it, and because it radically changed the way we study the brain. It's because half of the researchers was going to the brain. Thank you.
Okay, I heard the word optogenetics for the first time about a minute ago, so you're not going to get a technical question from me. You made an amazing claim that you can actually cure diseases with this concept and this technology, but are you not just playing with the symptoms of diseases like Parkinson's? How would you actually start to think about curing something?
Briefly.
Can you repeat the question once more?
It's the difference between the symptoms and the causes of the disease.
Yeah.
Because you use the word cure, which is a very powerful, strong word to use.
I know. For a disease like Parkinson's, the problem is the neuron is not working. So it's not secreting the neurotransmitter that allows it to work. So with this technique, we can actually, theoretically, we're not applying it. We applied it to some mice and stuff, and animals. But by applying this technique, we could actually activate the cell. Like when you're moving and when you're trying to turn.
Briefly.
Okay, when you're trying to turn on a car, it's the same when you're trying to activate the neuron. So when you're turning on the car, the tick tick tick, then it turns on. Thank you.
Okay, we have to turn your neurons off now. Ahmed Bara. So staying with ancient cradles of civilization experiencing a bit of modern turmoil, we go from Egypt to Greece, where a little under 2,500 years ago, the philosopher and proto-scientist Democritus came up with the forerunner of atomic theory, as you'll have heard if you were in the Sean Carroll talk about the Higgs boson earlier. Now, fast forward to now, and the largest research centre in Greece is named in his honour, NCSR Democritus. It's there that FameLab Greece winner Nikos Moustakis is a PhD student working in the field of solar fuels and artificial photosynthesis. When not working, Nikos tries not to let his fondness for video games get in the way of his efforts to learn to play electric guitar. And when not doing either of those, he says he feels good helping people distinguish good from bad science, which strikes me as just the sort of thing that democratic Democritus would have approved of. Let's see what good science we're able to distinguish after 180 seconds with Nikos Moustakis.
Let me tell you a story that happened to me a few days ago. I was wearing my clean and stylish clothes, and as I was walking down the streets of Athens like the new John Travolta, a taxi driver with an old rusty car stepped on the gas, and a big black cloud of smoke covered me and ruined my flawless style. I was angry, I was mad. I was so mad, an idea stuck in my head. What if we could use the carbon dioxide from the fumes of the taxi and transform it into something more useful rather than dispersing it in the environment? In that way, we could combat global warming and solve the energy crisis. Okay, I lied. The true thoughts about the taxi driver cannot tell you today. But my idea was correct, and I found out that scientists were studying this subject. But how are scientists going to do this? Of course, by copying nature and mimicking the procedure of photosynthesis. As plants use carbon dioxide, water, and the sun to produce their energy source and the oxygen, in the same way scientists are trying to use the solar light and the carbon dioxide with humidity to convert carbon dioxide into something useful, long hydrocarbon chains that can be reused as fuels, thus closing the carbon cycle. Unfortunately, the things are not so simple. So let's move to the world of comics. Imagine that a CO2 molecule, oh, thank you. A CO2 molecule is like an evil overlord that wants to take over the world. But he's stealthy because he has no odor or color. But the scientists are trying to find the ideal superhero to try to oppose him. The ideal superhero must be cheap, efficient, non-toxic, friendly to the environment, and in abundance. And these are only a few of the required qualifications to take the job for the superhero. One of the superheroes candidate or photocatalysts is titanium dioxide, as known as Titania. Titania, Mrs. Titania, as a female, likes sunbathing. So she takes the sun, the energy from the sun, and uses it to split water into hydrogen ions and hydroxyl radicals. These are her secret weapons, and they attack the carbon dioxide and forces it to combine with other CO2 molecules and hydrogen to make these long hydrocarbon chains that can be reused as fuels. These fuels are called solar fuels, and the whole process is called artificial photosynthesis. The truth is that we have a long road ahead in order to find this ideal superhero, but we are trying. Till then, if you come to Greece, and I hope you do, beware of the Greek taxi drivers. Thank you.
this is quite topical for me because we're voting on green energy and things like this, so and worrying about climate change. So on a scale of 0 to 10, in how easy it will be to actually make this happen, where 0 is sort of coal, which you can dig out of the ground and burn straight away, and 10 is nuclear fusion, which always seems to be 40 years away. How easy is this going to be to actually do?
The difficult part is not doing it. We are already doing it, but the efficiency is really too low. And we are trying, some labs are trying, like ours, we're trying to do titanium dioxide, which is friendly for the environment, non-toxic and cheap. Other labs are using gold or silver, which makes the photocatalyst expensive. So there's a balance between all the qualifications for the superhero to take the job. But the efficiencies are pretty low, and what we are trying to do is try to increase the efficiency to take, now we have ppms per liter, so of pure carbon dioxide into, let's say, methanol or methane or something that we can use as fuels to use electricity, let's say. So I would give him an 8.
8. Do you think it's better to use science in this way to sort of clean up the mess of things that are out there already, or would it be better to convince people not to use those technologies and invent cleaner technologies?
I think that we must use whatever we can to produce green technology, green energy, I'm sorry. If we can, as carbon dioxide is, we're dispersing in the environment, nothing to do with it, even that small amount of this carbon dioxide, if we could do something with it, it would be a great job. So.
Okay, I think that's a perfect point. He was graphic, he was novel. Nikos Moustakos. Now, last year, Ireland had their first ever FameLab competition. The winner came to Cheltenham to be part of FameLab International, and he won that too. The cheek of it, winning FameLab the very first time you've even held a competition. Now, as anyone who knows anything about Irish culture knows, or certainly if you've watched Eurovision or Father Ted, once Ireland start winning things, they have a habit of finding it very hard to stop. So, going for an unprecedented, un-Taoiseach Irish International FameLab double whammy, we have Aidan Murphy, a PhD student at Dublin City University. Like a Bond villain, Adam spends his time surrounded by lasers and chemicals and gold, but rather than trying to take over the world, his goal is to develop nanoscale platforms to help detect diseases, then to take over the world. He loves squash and sci-fi and talking physics, and tonight he gets to do at least one of those. A sizable hand, please, for the only 2014 FameLab Ireland finalist to wear a hat, Adam Murphy.
Could you all do something for me? Take yourselves back to when you were kids and the wonder and the awe you felt the very first time you saw a magic show. Now let's all get a little bit older and realize the disappointment you felt when you realized it was all a trick. A mirror or a particularly bendy lady. The moment you realized that none of us are getting our Hogwarts acceptance letters. Well, I've had enough. I am going to show you some real magic tonight. With the help from a little bit of quantum physics, I am going to bend the fundamental speed limit of the universe. I am going to bend the speed of light. But I'm going to do that with something called Schrodinger's cat. It's a thought experiment about what happens when you put a cat in a box. Now, I don't have a cat, and I don't have a box, but I do have a hat, and I do have a rabbit. So, we put the bunny in the hat, and in with the bunny, we put a vial of poison that has a 50-50 chance of opening and killing the rabbit now. So I don't know if that rabbit is alive or dead. There's a 50-50 chance of either. So according to quantum physics, the rabbit is alive and dead at the same time. It's a zombie bunny. So what happens if we add another hat and another rabbit? So over here, we put this bunny in this hat with a vial of poison that's different. If that one goes off, this one doesn't. If this one goes off, that one doesn't. You'll take care of him, won't you? Thank you. So now, this rabbit is alive and dead, and that rabbit is dead and alive. Let's see how they're getting on. So I look in here, and yay, the bunny lives! But because they have to be opposite, I'm now a horrible bunny murderer. But despite this, something remarkable has still happened. When I see that he's alive, he instantly becomes dead. It wouldn't matter if that hat and that rabbit was in Dublin. It would still be instant. It wouldn't matter if that rabbit was on the other side of the universe. It would still be instantaneous. It would be faster than light, all because the fates of the rabbits have been entangled. Quantum entanglement was put forward by Einstein as a way to knock quantum mechanics. He called it spooky action at a distance. It was an Irishman, John Stuart Bell, who proved that it was possible in the 60s, and since then it's been used to do amazing things, including just last week when a group of Dutch physicists used it to do something truly incredible. They were able to take entangled electrons and teleport them 10 feet away. What Schrödinger was trying to say with all this is we don't have zombie bunnies hopping about the place. Where's the line between our world and the quantum one? And if you ever figure that out, let me know and we'll go to Oslo and split the Nobel Prize money. But it does illustrate my favorite thing about science. Magic tricks lose their luster when you know how they work, but science just gets more impressive. Thank you for listening.
Thank you. I hate magic, but I love physics, so that's good. I like your message. Can you just explain again, I understand the sort of instantaneous travel thing of one being alive, one being dead. How does that equate to what they did with the light transportation of the photons, the teleportation? Just sort of that
the
connection.
group last week, they teleported electrons.
Yeah.
So what they did, electrons have this interesting property. Like the Earth goes around the sun, but spins itself, electrons go around atoms, but spin themselves. And they can spin in one of two ways. They can either spin up, or they can spin down. But because these are quantum particles, they can do both. They can be spinning up and down at the same time. So 10 feet away, they had these two electrons that were going about their business, not really doing much. And then they started pumping microwave pulses at them. But the two labs knew what microwave pulses they were sending. They weren't measuring them, they were leaving it to the computers. But they were putting both of these electrons into an entangled state by knowing what microwaves they were putting at them. And then they started pumping the Jesus out of them with a laser. And then every so often, the two electrons would release particles of light, photons, in the same direction. Now the thing about these, spinning that way releases a different color of light to spinning that way, say red and blue. It's actually blue in a very, 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 very different shade of blue, but we'll say red and blue. And then they went off and they bounced, and it was kind of like, we don't know which photon's going where. We don't know. Now which one do we have? So then they measured, and they saw, oh, the blue one. Where did that come from? Oh, that came from that one. So they knew what way that one was going. And because they knew what way that one was going, the other one switched. And then all they had to do was knock that one back, and suddenly they teleported these electrons 10 feet away. It's incredible physics.
Very quick second question for Adam, because no answer should be longer than the presentation.
I've got one. I mean, this is about observers making a difference.
Yeah.
Does it have to be a human observer, or could it be another bunny?
It could be another bunny. The reason Schrödinger's cat doesn't apply is because the bunny can think, and this is the most morbid sentence I've ever said, the bunny knows it's dying. Any measurement, just a machine would be able to make this difference. It's a measurement that changes it, not a human observer. It's really cool.
Okay.
Take my bunny back.
Pump your hands like the Jesus for Adam Murphy. I will now be unable to think of Einstein talking. It will now be spooky action at a distance with a little dance move in there as well. Just before, why do you hate magic so much? Just very quickly.
I want to know how it works. It frustrates me immensely.
Okay, fine. I love magic. Anyway, next to the country currently ranked 17th in the world in ease of doing business index, and where, in my experience, a disproportionate number of people love playing board games, which is fine by me, because I love them too. I am, of course, talking about Lithuania, and their 2014 champion is Marius Polianskis. Now, Marius studies medicine, and he's a particular fascination with odd and unexpected interactions between bits of the body that don't seem to be related, like your eyebrows and your spleen. I'm not saying there is an interaction between your eye, but those are just two bits of the body that don't seem to be related. A better example might be kidney and ear, or perhaps between FameLab audience and Marius Polianskis.
Life is quite boring. That's why so many people turn to soap operas in quite rainy evenings. But you don't have to look far to find a fascinating story, because one is happening right now in your own body. So please meet the cast of your soap opera episode. The first one is all-beloved, simple man, an organ in your abdominal cavity that is filtering your blood 24/7. It's, of course, the kidney. Second lead role belongs to an organ which is usually seen with a fancy jewelry. And it's the ear which allows you to hear my slightly trembling voice. Our two main characters are very far away from each other. Kidney and ear, they look very different from each other. It seems they have nothing in common. Or do they? Following the example of most soap operas, our hard-working kidney and jewelry-fond ear has this special connection. When you look under the microscope, a part of inner ear called stria vascularis is very similar to kidney tissue. Moreover, they have similar functions, like they transfer water and electrolytes, which is important for proper hearing and production of urine. Our two main characters have to face tough challenges along the way. There are certain drugs like antibiotics, which present side effects like impaired hearing and kidney dysfunction. In addition, there are like genetic diseases like Alport syndrome, which cause deafness and kidney insufficiency. So our two main characters are so simple in all these ways: structure, function, drug toxicity, and genetic diseases. But you'll be surprised to hear that they are even more interconnected. Sometimes when primary kidney insufficiency results in hearing loss, scientists believe that this happens because of a chain reaction. Kidney insufficiency results in disbalance of blood electrolytes, which in turn results in high-frequency hearing loss. As you hear the rain intensify, maybe you think you have high-frequency hearing loss, but it's not correct. It's hard to find high-frequency hearing loss in everyday situations, but diagnosed high-frequency hearing loss tends to progress till the point when a person needs a hearing aid. So, with current diagnostic devices, we can detect early high-frequency hearing loss and then try to treat it or at least slow down the progress of it. So possibly, this special relationship between our two main characters, the kidney and the ear, may help you in the future to hear your favorite soap opera much better and clearer. Thank you very much.
This is a fascinating story. So, are you actually saying that the ear and the kidney are connected, or it's just they have things in common?
Well, in English language terms, maybe you are correct. They have things in common that influence both of them. So, yeah.
So it's not that if I have a kidney transplant, I'm going to hear differently?
No, there is no wire between your kidney and the ear, so you don't have to worry.
Okay.
So is it just something that changes about the blood? I wasn't quite sure which part of the ear you meant was similar.
It's a part in inner ear.
Is it in the cochlear part?
Yes, that's right.
Okay.
It's in the scala media, the middle stair, if I translate it correctly. Yeah, it's the disbalance of the electrolytes in the blood.
Okay.
The current theorema says that.
So it would also be a link to any other part of your body that's... Is there any other parts of the body that would also be linked in the same way?
Yes, of course.
Okay.
There are many of them. Like Quentin said, an eyebrow and the spleen can have a connection, just we have to keep trying to find it. And that's why science is so great.
And you explained very clearly how you can sort of learn something about diagnosing hearing loss by looking at the comparison. Is there anything you can learn about the kidneys from the ear?
Oh, yeah, of course. One, like when we study medicine, there is like a smell of acetone. So if your kidneys don't function properly well, I could smell an acetone from like here, from you. So in ancient times when we didn't have so many and good treatment methods, kidney ward smelled like hell. Sorry.
Okay, please link your brains and your hands to applaud Marius Palanskis. And I'm not suggesting the eyebrow spleen line of research. Now, as I mentioned earlier, not only is the current international FameLab champion from Ireland, but once the Irish have won something, they have a tradition of winning it repeatedly. And what might just help them do that would be if they could somehow find a way to get Irish people to win more than one FameLab. And in fact, they do. As well as Ireland itself, another 3 countries tonight are represented by an Irish winner. Now, it's not quite as bad as it sounds. FameLab Benelux is unique in that whereas all the other competitions are for individual countries, this one, and the clue is in the name, spans Belgium, the Netherlands, and Luxembourg. And yet, with the talent of 3 great nations to call on, they still managed to have a winner from Ireland. That worthy winner is Pádraig Flood, appropriate name for this evening, who after a degree from Dublin is currently finishing his PhD in plant genetics at Wageningen University in the Netherlands. I've been practicing that all day. Pádraig's research focuses on using our understanding of plant genetics and natural variation to make them easier to grow and tastier. When not doing this in the lab, he's doing it al fresco as he loves eating wild plants wherever and whenever he can find them. I'm pretty sure I saw some fennel somewhere out there in Imperial Gardens, but no nibbling until after we've immersed ourselves in 3 minutes of Pádraig Flood.
Between 2.5 and 3 billion years ago, something happened which forever changed the fate of the Earth. It is the reason the sky is blue, the land is green, and our blood is red. It gave rise to the current chemistry of the atmosphere and oceans, and not only that, it turbocharged life itself, powering it to places it could never otherwise reach. But the most profound thing about this event is that it happened only once. Evolution is incredibly adept at finding optimal solutions to complex problems. Think of eyes, wings, and even intelligence, all of which have evolved multiple times. But this event, this innovation, evolved only once. So what was it? Well, small bacteria called blue-green algae hit on something amazing. They developed a way to split water and to use the resulting electrons and protons to capture carbon dioxide and make sugars. And that wasn't all. As a result of splitting water, they excreted the byproduct upon which our very lives depend: oxygen. This combination of sugars built with the sun's energy and oxygen providing the means for other organisms, such as ourselves, to extract this bottled sunlight gave life an unprecedented boost. As oxygen accumulated in the atmosphere, new life sprung forth, and for the first time in the history of the planet, complex multicellular organisms appeared. This process of splitting water and capturing carbon dioxide is called oxygenic photosynthesis. It is the most complex, energy-transducing process in biology, and it is dangerous. Splitting water within a cell is a very delicate undertaking. One mistake can be disastrous, leading to massive cellular damage and ultimately death. And yet, plants and algae do this on a daily basis with the most basic ingredients. So I think this process holds within it a promise for our future. These ancient bacteria managed to harness the might of the sun and generate hydrogen as a fuel. When burned, the waste is water. This at once solves both our energy and our climate crisis. Our salvation may be written in the genes of a lineage dating back more than 2.5 billion years, but only if we can figure out how to repeat it. Because remember, this evolved only once in the 4 billion year history of life on Earth. If we were to rewind the tape of life and play it again, would photosynthesis evolve? If life has sprung forth on other planets, how often will it solve this puzzle? For without this, the planet will never generate ozone, and over time, its oceans will be stripped away by ultraviolet rays, much like what we see on Mars today. So with your next breath, I want you to think of those ancient bacteria and how lucky we are that they cracked this puzzle and brought light to life. Thank you.
So, you're saying that blue-green algae have done more to protect our environment than the coalition? That's shocking.
Well, they've done more to change it, that's for sure. They've certainly done more to change it.
Can I just ask? You're very clear that this happened only once. How do you know?
Very good question. So there are multiple lines of evidence. Like everything in science, there's not just one thing that we can base our answer on. And in this case, I think the most convincing piece of evidence is if you look at all photosynthetic organisms, the machinery that they use is the same at its core. It's all built of the same building blocks, arranged in very similar ways. The chances of that happening on multiple occasions and converging on this identical structure is very, very low. We also see genetic convergence. We see convergence in things that they do which don't necessarily make sense. So we're very confident. It's one of those facts in biology that we're extremely confident about, that this only happened once. And for me, that is amazing, because there's even some scientists who think that life is more likely to evolve than photosynthesis. And without photosynthesis, none of this would be here, because it just completely changes how a planet is. It gives it ozone, it gives it oxygen, it gives a completely different chemical balance, which allows multicellular organisms like us to exist. Otherwise, we just wouldn't be able to extract enough energy from our food, because anaerobic respiration is just not good enough for us to be able to do what we do. Think, our brains need oxygen, you know?
You had a beautiful poetic start about this being the reason why the sky is blue, the grass is green. Why exactly is it the reason why the sky is blue?
I didn't have time to explain that. I apologize. So the oxygen is blue. If you take liquid oxygen, it's blue. And nitrogen is transparent. If you take liquid nitrogen, it just looks like water. So the main constituents of the atmosphere are oxygen and nitrogen. So that oxygen in the atmosphere gives the blue skies. Before that, it was probably red from CO2. So we would have red skies. So that nice sky blue comes from the oxygen. The green comes from the chlorophyll that these organisms need to capture sunlight so that they can split water. And the red in our blood, just to finish, comes from the fact that the iron is oxidized. So you know you have two colors of blood. In your veins, you have like a dark purple blood, but in your arteries, you have this bright red blood. The fact that it's bright red comes from the fact that that iron is oxidized. So without that oxygen binding to the heme, to the iron in the heme, you wouldn't see that color.
Red-blooded blue sky science from Poric Flood. And after the mention about the blue-green algae, you're not suggesting that a coalition between the Conservatives and the Greens is the way ahead for the, to save the environment, are you? No, I didn't think you were as well. Okay, halfway, and next is our current German champion, Anna Stuckel. She's another example of the have talent, will travel approach of many FameLabbers. She's doing a PhD at Lund University in Sweden, but she came back home to Germany to take part in FameLab. Anna is a neuroscientist with a particular passion for putting herself inside the heads of animals and trying to understand how they see their world. To that end, she apparently spends a lot of time making insects watch television, which got me wondering what their favourite shows are. The best I could come up with was Desperate Houseflies, Beetlestar Galactica, and Insects in the City. Plus, of course, anything on the BBC. Okay, apologies. And some of those were very culture-specific. If you've got better ones, tell me during the interval. Now, when Anna's not unfairly boosting viewing figures, she says she likes to get as much sport as she can fit into one day. But how much science can she fit into 180 seconds? We're about to find out. Bang your mandibles and antennae together for our FameLab winner from Germany, Anna Stuckel.
Now, welcome the Queen of the Night. Master of seeing in the dark, who can still fly around when the sky is only illuminated by stars. I'm sure you guessed it, I'm talking about a moth. And how this particular moth, the elephant hawk moth, can see so well at night. But first, why is it so hard to see when the light is very dim? Follow me on a journey into the eye where we meet the light detectors or photoreceptors. Their job is to detect light particles, the photons, and if they do their job well, we can see well. During the day, this is no problem. There are tons and tons of photons. The photoreceptors are literally standing in a massive shower of rain, or photons, basically like this, and you could just feel that no problem detecting raindrops or photons. During the night, however, there are only very few photons. And the poor photoreceptors feel a bit like you would when you're out on a gray day and you get hit by a raindrop, and then nothing. And then you start wondering, did I really feel this? or did I imagine it? Is it raining now? So seeing at night is so hard because there's so few photons drizzling in, and the photoreceptor's detection is really unreliable. But how does my moth solve this problem? Well, according to the principle of the great scientists, the Beatles, I get by with a little help from my friends. Because I'm investigating nerve cells in the brain of this moth that get help from their friends. They're reaching out and collecting information from their neighboring cells about the photons. It's a bit like when you're out with your friends when you get hit by the very raindrop. What do you do when in doubt? Well, you turn around and ask, hey, did anybody else feel something? And if only one or two of your friends felt something, you can be sure you didn't imagine it, but there was actually a raindrop. So you improve the reliability of your initially unreliable detection by getting information from neighboring entities. We call this principle spatial summation, and this is what my moth is doing in her brain to improve vision at night. Now, maybe some of you ask yourselves, who on earth is interested in moth vision? For example, companies who develop cameras to help us drive our cars. They should work at night, and they can actually use our findings from the moth brain to implement in the software of the cameras and thus benefit from the tricks of the queen of the night. So remember, life's easier with a little bit of help from our friends, and so is vision at night.
Okay, I'm loving the singing. I'm loving the singing. I'm sold on the singing. That's it. You know, tick, thank you, done. So it's not the eye. I always thought moth's eyes, you know, the whole thing, you know, the big, you know. But then clearly it's not. You're saying it's all in the brain.
I'm studying what's happening in the brain. There's certainly a lot happening in the eye already.
Just do the brain thing again, because I don't think I've quite got it. How does it work? And can I do it with mine?
Well, what it does is basically that if you have a very unreliable detection or signal, you get information from your neighbors who also detect this signal. And by getting, so to say, more samples of this, you get a better or more reliable detection. And that's what happens in the moth brain. So individual neurons reach out to neighbors who also try to detect these photons. And by getting this information from the neighbors, they might improve the detection. So maybe they don't get the photon that comes, but the neighbor gets it, and so they get the information about it. And that makes it more reliable.
Is it specific to moths? Or
no,
other?
But it's very well to be studied in moths.
And what other species have it?
Well, well, first of all, I think actually humans have it to some degree, but in their eye. So the ganglion cells are also, especially in the periphery of your eye, reaching out to the neighbors. And I'm sure a lot of other nocturnal insects have it. It's just a matter of studying it. And we picked the moth to study it, so...
Poor moth.
Yeah.
So with the application in cars, I didn't quite understand. Is it to do with cameras taking lots of images and how it synthesizes them together? Is that how it...
Not necessarily. Basically, what you can do is... Cameras have, well, the resolution is measured is pixels. Yeah. And what you basically do in cameras then is making bigger pixels. Hopefully in a smart way, just don't make them bigger, but you actually use smart processing that you can form after what we learned from the moth brain. But eventually making bigger pixels, that's what you do.
Okay, bigger pixels for our mathematical genius or mathematical genius, Anna Stuckel. And an extra round of applause for Godzilla's friend Mothra there, making a special appearance. Yes, because you had props, you had a musical number. It's what you wished. They're all going to start trying to work songs into their remaining routines. Now, after an Irishman representing Belgium, Luxembourg, and the Netherlands, and a German based in Sweden, we now come to our French FameLab winner, who is from California. Now based in Paris, David Davila studied what he likes to call all things brainy. You didn't think it could rain any louder, but it can. But the rest of us call it neuroscience. When not studying all things brainy, he likes to talk about it to his friends or share stories about it online as co-host of the Brain Drain podcast. And when not researching or chatting about or podcasting neuroscience, he does FameLab. Get those neurons firing for our non-French France FameLab winner, David Davila.
This is one of my favorite songs. It's simple, but still moving. And that's really what makes this song good, isn't it? When it moves us, when it makes us feel. But how is it that music, the patter of rain, what is essentially noise, can move us to our deepest emotions? Well, as a neuroscientist and self-proclaimed huge nerd, my instinctive response is to ask, well, what's going on in the brain? What brain areas are responsible for the emotions we feel when we listen to music? They only gave me 3 minutes, so I'm just going to focus on positive emotions. But even then, there are still 2 different brain areas to talk about. There's the hippocampus and the nucleus accumbens. Now, the hippocampus has been known for a while to be involved in positive emotions because we find in depressive patients, it's a lot smaller. And with antidepressive treatments, it gets bigger. So it should be no surprise that activation of the hippocampus is implied, linked to quote-unquote tender, positive emotionality when listening to music. That is to say, some evidence suggests that the hippocampus's activation is what's responsible for the warm, fuzzy feelings we get when we're listening to some music. The other relevant part of the brain is actually one of my favorite parts of the brain, and it's called the nucleus accumbens. It's part of the brain's reward system, and thus is heavily implied in feelings of pure pleasure, sex, chocolate, cocaine, everything else we love in life. But in particular, the nucleus accumbens is responsible for the fun feeling that we get of music. If you've ever liked a mindless, silly song just because it was fun and stupid, it was your nucleus accumbens bringing you back for more, even though you actually hate One Direction. Remember that these are actually 2 very important brain areas, and they're not just there to respond to music. In fact, the hippocampus especially is dysregulated in mood disorders like bipolar disorder and depression. And thus, music therapy is actually a booming field because we're starting to realize that listening to music has a physical effect on our brain. I don't know how close we are to treating depression with music. In fact, maybe until we understand the effect of music completely, it's best to sit back, listen to some nice music, activate our happy areas, and relax, unless we have some chocolate and cocaine on hand. Thank you.
If you've got one bit of the brain that does sex, chocolate, and music, I wonder why you ever evolve the rest of it, really.
Just throw it out.
The One Direction question is quite interesting, because my daughter really likes One Direction. Why does her brain produce such a different emotional response to my emotional response to One Direction, which is near suicidal?
That's hormones, isn't it?
Well, this was a talk about music and not about cute teenage boys. And that's a visual effect, not an auditory one. But the question's valid.
Yeah.
Why are some of us addicted to some kinds of music and some of us are completely averse? And the answer I have is that I do not know. I don't know if anybody. But the question is similar as to like, we all have the same taste buds. Why do some of us hate broccoli? Some of us love it. That's actually a lie. We don't have the same taste buds. But the same kind of thing.
You can answer that question if you can't answer the other one.
In fact, it's a very complex system, and there's no real simple way to explain why our experiences or how our experiences morph our preferences, whether genetics plays into a role. It's all very, very spread across lots of different factors. That is too much to squeeze into one brain talk.
Do we know how much the effect lasts after the music stops? So if something's happening in the brain when you're listening to the music, can you find out, can you see or can you measure in any way how long it lasts when it's finished?
It really does depend on if we're talking about this fun cocaine effect, it's really short. You can kind of feel that almost. You like the song, you hear it, it's over, you're done. If you listen to music that maybe is more related to our mood, our hippocampal activation, then you can imagine that lasts much longer. In fact, you know, you listen to a good song, you're feeling good, it may stay with you for the rest of the afternoon. I'm not going to say there's been any studies on exactly how long it will affect you, but it does depend on what exactly you mean by the positive emotionality.
So if it's more emotional rather than a quick fix type song, you mean it might?
It
be
might,
more
yeah,
effect?
it might be, yes.
Okay.
Not just a One Direction one. Any campus would be a hippocampus with our new romantic David Davila. Glad to see David took his brain with him. Now, FameLab, as you've established, is a global competition, but we do have one sort of local finalist, our UK winner, Caroline Shenton-Taylor. I'm not sure she can help Porrick find edible plants out there, but should you ever need to identify a tree on a dark night, Caroline is the woman for you. She's taught people how to recognise bark blindfolded. She's also explained juggling using Venn diagrams, or possibly Venn diagrams using juggling, I wasn't clear, and sent small children into raptures by getting them to send water-powered rockets into the stratosphere. But mostly, Caroline's worked on the deep structure of materials using powerful X-ray sources, superconducting magnets, and ultra-low temperatures. Her name is an anagram of an erotic loony enthralls, so stand by to be enthralled, probably not erotically, by Caroline Shenton-Taylor.
Next year, you might be able to buy the coolest new kitchen gadget. The humble fridge has been on a quiet scientific journey, one that's taken over 100 years and required us to understand a strange magnetic effect. But by doing so, we're on the verge of having a new fridge that's safer and greener. Now, it all started back in 1881, when a scientist called Emil Warburg noticed that if you put a lump of iron near a magnet, it got a tiny bit warmer. And if you took the magnet away, the iron, it got a tiny bit colder. But at the time, it was just a bit of a scientific curiosity. It took us another 40 years to work out what was going on. What he'd seen was the magnetocaloric effect. So if you imagine a lump of magnetic material, you can think of it as having a lot of little bar magnets inside, each with their own north and south pole. Now, originally, all these bar magnets point in any direction they want. But if you put them near a strong magnet, they all line up, and you get an ordered state. Now, the neat bit is if you take that magnet away, the little bar magnets want to go back to being randomly orientated. But to do that, they need energy, and the energy that's available to them is the heat within the material. And so as they move, they suck up the heat, and the material gets colder. Finding that we could use magnets to make things get cold was a breakthrough. And in the 1930s, we started to think, well, can we do something useful? Can we make a fridge? But we could only cool things by a degree, which isn't going to keep your beer cold. The breakthrough came in the 1990s when scientists discovered gadolinium-based compounds. Now, these compounds don't just have the magnetocaloric effect, they have a giant one. When you move them in and out of a magnetic field, their temperature drops significantly. And that was the point that the fridge became a reality magnetically. Next year, you could go into a shop potentially and buy a magnetic fridge. And if you choose to do so, you won't have the chemical coolant that you currently have in your model at home. But even better, the scientists reckon you might be able to save up to 40% of your energy bill. However, I think there's actually a deeper message to this story, because lots of the technology in our home is based on science that came about because of a curiosity. And I think it's when we fund this curiosity-driven research that we can look into the weird and wonderful materials today that we might not understand, but could be the technology of tomorrow. So whether you choose to keep your magnets on the outside or the inside of your fridge, I think the magnetocaloric effect shows science, it may take a while, but it really does lead to cool stuff. Thank you.
I was burning to ask at the end whether fridge magnets would work on it, and then you stole the line. Really good. Wow. Are there other discoveries that you feel the same way about that have kind of happened serendipitously in science?
Yeah, there's a few. So I think orthotic materials, they're really crazy. They're materials that we kind of discovered by accident. When you stretch them, they get fatter. Didn't really know what to do with them. Then in the 1970s, we worked out we could make them. And now we're using them in body combat armor. So when the bullet hits you, the material gets fatter and it protects you. So I think that's cool. So yeah, there's a few.
Oh, this is a bit of an obtuse question. I'm sorry about this. But it's, in MRI, when we're doing MRI scans, we inject gadolinium into patients. And they go inside a big MRI scanner. So are they, as the stuff goes through the patient, are they getting hotter and colder? I mean, is it the same? Could this effect be going on?
Potentially, but because you're using, I think, is it neat gadolinium you're using? I think that's different. So the gadolinium I'm talking about is mixed with like palladium or with silicon and germanium. And I think it's... I said gadolinium compounds.
You didn't say that.
But I think it's the mixing that magnifies the effect, I think. And I think when you've got it neat, you get a tiny effect.
Right.
Well, I hope so for anyone who's been in a machine.
And so is this thing, this fridge, this magneto fridge, is it real?
Yes.
I mean, you really can.
Well,
you can't buy one, but
next year, potentially you can buy one. So the scientists have been working with companies. Obviously, companies are saying it's going to be really energy efficient. The scientists are being slightly more reserved. But yeah, within the next 12 to 24 months, the stories are saying that it should break and you should be able to buy one.
So how big a magnet and how much stuff do you need to sort of waft around? Is it like the size of a small house?
No, it's better than your current fridge. So your current fridge has got that big compressor on the back, which compresses the gas to the liquid to circulate round. Yeah. Don't need that. You can have a nice slimline magnet at the back. And it'll also be really good in planes, they reckon, because it's a solid material rather than a liquid gas system. I'm not on commission.
Sorry, I'm going to have to overrule you. I'm being Mr. Speaker here. Thaw your hands out by applauding, please, for our refrigerator orator, Caroline Shenton-Taylor. Sorry, has to be the same amount of questions for everyone. And Mark, point of clarification for you as a judge, asking whether it's real. They're not allowed to make stuff up. Okay, that's an important detail you should grasp. Into the final furlong now, and there is no horn of doom has been used all the way through so far. And we have our two furthest flung FameLabbers coming up. First, our Hong Kong champ, Ding Li, a Masters of Student of Environmental Engineering and Management in Hong Kong University of Science and Technology. She's come over 6,000 miles to be here, although it won't faze Ding Li. As well as being a voluntary zookeeper in Hong Kong, she's also spent time as an exchange student in Australia and a school teacher in Ghana. She says she enjoys annoying her friends with nerdy scientific facts, and that what's great about FameLab is she can now do that in public. I'm not sure that being annoyed by a nerd is entirely how I define FameLab, but let's see what mood we're in after stepping into the ring with Ding Li.
What am I doing now? I'm smiling. How do we smile? First, we contract our zygomaticus major, our orbicular ocelli. Then we smile. Smiling is a universal nonverbal expression of all humans. We're born to smile. But actually, why do we evolve this weird gesture of lifting your corner of your mouth up to communicate? It's actually because our primate ancestor used to do this to prove that, to tell the others that we're friendly, we're not harmful, no trouble, please. So people nowadays, like human, we smile to tell the others that we're friendly. So how do our brain tell us to smile? We first of all secrete a neurotransmitter endorphin trigger us to smile. But then when we smile, we get back to our brain and stimulate our brain reward system. This reduces our stress hormone level like cortisol, adrenaline, increase our mood-related neurotransmitter like serotonin and norepinephrine. And the most important thing is actually increase our happy hormone level, dopamine secretion. And 1 minute ago, I say dopamine stimulate us to smile, and when we smile, we further stimulate our endorphin secretion. So it's a positive feedback loop. And this is actually proposed by Charles Darwin in 1872, which is known as the facial feedback response theory. In other words, when your brain feels happy, we smile. And when we smile, our brain feels happier. So when you don't feel like you're in the mood, you can just smile, because your brain will think you're really actually happy and will be happy. But if you really don't want to pay that effort in faking a smile in front of the mirror like an idiot, what you can actually do is be with someone who smiles. Because smiling is evolutionary contagious. What I mean is, a study shows that when we look at someone who smiles, it's really hard to put on a long face when they're smiling at you. Because when we see people smiling at us, we actually stimulate our brain mirror neuron, suppressing our controlled in facial muscles, and stimulate our own smile. So you smile, I smile. Yeah. And apart from the happiness that bring to smile, that happiness level to bring to us, smiling has actually numerous health benefits. It reduces depression, reduces anxiety, reduces blood pressure, reduces blood rate, and even reduces pain. And the study shows that one smile is estimated to trigger our brain happiness level equivalent to 2,000 bars of chocolate. So if you want 2,000 bars of chocolate with those calorie incurs, what we actually do now is smile.
So are you seriously telling me that all I have to do to go back to the House of Commons, or we go to Legco in Hong Kong, to get them to get on, is just tell them to smile at each other a lot? Are there practical applications to this?
You mean when we smile, do we have practical application?
Yeah, I mean, could you really reduce tension in conflict situations?
Of course we do. Like,
Without
I'm
looking
smiling
a bit
at
like an
you, you feel like I'm more friendly.
He's immune.
Yeah. Yeah, of course we do.
Can I ask then, does it make a difference if it's of just a picture of someone smiling rather than a human in front of you smiling? Can people tell the difference? Does it have a different reaction if it's a fixed picture?
Actually, it's no difference. Because there was a study that shows that when you look at someone who smiles, you can't tell they're fake or real smile. But then actually our mirror neurons kind of mimic that smile. And then you know it is real smile or fake smile. It's actually looking at the picture of the people smiling. So it's actually the same. It's the same thing. Because we interpret people in the picture and real person the same way.
So does anyone actually using this to treat people with depression? Is it as simple as being able to just make someone smile more?
Actually, for depression patient, it's not really about what you smile or not, reward system or not. They have some kind of abnormal secretion of hormones.
Okay.
So actually, yeah, of course, smile could actually help that, of course, but it's more than that. It's not just smile, it can treat depression, but it can reduce depression.
Okay.
Yeah.
So it gets more complicated because with smiling, you can get laughter as well.
Yeah.
You know, I can be smiling with you but laughing at
And
you, or I could be laughing with you. You know, and so why did we? Where's the laughter? Why did we evolve laughter? What's that? Where does that come from?
Actually,
where's the laughter side of it?
Laughter is just another level of smiling, but it's not the same mechanism as just smiling. Actually, I'm not sure about why we ha, but...
Okay, we need a bit of aha about ha-ha. So complete the positive feedback loop, please, for the Born to Smile, Ding Li. And I know this is a scientific event, but having mentioned we hadn't had to use the Horn of Doom, I clearly jinxed it there because we had to use it that time. So penultimately, we come to the finalist who has made the Ultima Thule journey well over 10,000 miles from down under to up on this stage. Our first ever FameLab Australia winner, Dr. Michael Smout, is a Brisbane-raised, Cairns-based geek since the age of 6, who is now a parasitologist working with the Queensland Tropical Health Alliance. His research has spanned melanoma and childhood viral infections, but, as he puts it, through a convoluted series of wonderful events, he now loves all things wormy and toxic. And he wants to share that love now. A very warm welcome, please, for the reigning Aussie FameLab champ, Michael Smout.
I want you to imagine you're in your bathroom, you slip over, you fall, you hit yourself, maybe even cut yourself. What if you had magic bandages that could supercharge that healing? Heal it right away. Well, I'll get to that in a minute. Who likes to travel? Who's been to Southeast Asia? Are you one of the 800,000 Britons who go to Thailand every year? Well, maybe you need to be a little bit more careful. If you like to eat the local cuisine, if you've eaten raw fish, maybe you've brought home a friend. Teddy's been to Thailand. He loves to travel. He goes to all the remote places. He eats some raw fish. Unfortunately, he brought home a friend as well. But luckily, I'm a doctor. I can help. This is Teddy's liver. Teddy brought home Larry the liverworm. Now, unfortunately, the ones we can catch aren't quite as cute. They're about the shape of a leaf and the size of half a matchstick, and you can get hundreds if you eat a lot. What's the problem with getting the liverworm? Well, we've found that it secretes a growth factor that supercharges that wound healing. So what's it doing in you? Well, it's munching around your liver, causing wounds, then healing them up, causing wounds, healing them up. Now, in the short term, this is great. It wants you to be nice and healthy. It's going to live for decades. But over the long term, over the decades, it eventually can cause damage. And that eventually can lead to liver cancer. And as you can imagine, that's a problem. This particular type is deadly and kills within a year. As a tourist, it's not particularly a problem because you can take a pill. That'll get rid of it. And don't worry, your sushi's fine. But where it is a problem is for the 900 million local Thais who have this disease, and 20,000 of them die every year. So we're working to use this growth factor to create a vaccine to help these poor, impoverished Thais. But what does this mean for those of us not infected with the worm? Well, by understanding this growth factor, we believe we'll be able to supercharge our wound healing. It won't give us Wolverine-like superpowers, but it may create super bandages. We've got evidence that by adding this growth factor to a gel, we can double the speed of wound healing in just 2 days, which is particularly handy, especially when there's large groups of people, the elderly, diabetics, and military personnel, who have wounds that just don't heal. So we believe by adding it to bandages or adding it to gels, we'll be able to supercharge their wound healing and help these wounds be able to heal up properly. So to summarize, don't eat raw fish in Thailand, worm spit causes cancer, and magical bandages may be just around the corner. Thank you.
I've heard they use maggots in wound healing. Is it different to that?
That's more getting rid of the bad tissue, but that is part of it. I'm not sure whether they actually have much of a factor. I think that's more removing the badness, whereas this is actually helping healing this is maybe more in ulcers rather than wounds in that sense.
Okay.
So I keep hearing that the magic bandages story, and there's loads of wound healing, technologies out there, and molecules they're finding the whole time. What makes yours special? Because this story's been around for a bit, and if it really worked, we'd all be getting our elastoplast and just sticking them on and everything would be fine. Clearly, it's not that easy, is it?
No, it certainly isn't. And our work's still very early. We've only come up with this a year and a half ago. I've been working on this for a decade on more the cancer side, but finding out how it will be able to help us is a new angle. And certainly anything like this needs to go through a lot of testing to actually become a product.
So how does it do the wound healing? And will it heal, like you slice a brain, will it heal the brain, or does it do just specific tissues?
Short answer, I don't know. Long answer, probably. This growth factor is very similar to what we use. It's actually granulin. It's what we use in our wound healing. And despite they've been known about it for 2 decades, there's been very little work in actually finding out how it's doing what it's doing, actually because it signals so many different ways that it's really challenging to work on. And so most likely it would help pretty much everything because it's a very universal growth factor found within us.
Of course you're going to tell me that.
For the layperson, apart from saying that Wolverine was my favourite X-Men, what is a growth factor?
Anything that causes your cells to change their number, generally increasing or change their growth. So, for instance, if it causes one cell to turn to 2 cells, that's causing growth.
In this case, what is it?
That's exactly what it is. It causes your cells actually to go into hyperdrive. and just replicate many, many times faster than they normally would, or not at all as in a normal situation.
A tricky one. How do you create a vaccine out of a growth factor then?
Probably
And a very short answer as well.
Probably by chopping it in half so it doesn't have its growth factor capabilities, but can still create a vaccine.
Okay, he brought on a teddy bear, a giant worm, and a velvet liver. One gift for each judge. Please thank the gifted Michael Smout. And people of Cheltenham know that your MP dreams of having adamantium claws. And finally, that's for Marvel fans out there. Now, and finally, or semi-finally, to polish things off, we have our Polish winner, Dr. Joanna Bagniewska. A zoologist, she specializes in behavioral ecology and biotelemetry, which she defines as trying to figure out what animals are doing when no one is watching, which sounds a bit less intrusive than making insects watch television. Now a lecturer at Nottingham Trent University, when not sneaking up on unsuspecting mammals, Joanna spends her time making things out of beads and taking part in ballroom dancing competitions. It takes two to tango, but it only takes one to FameLab. And that final one is our Polish champion of 2014, Joanna Bagniewska.
Who here feels like they need more sleep? But would you be so keen on taking a nap, gentlemen, if I told you that sleep could shrink your testicles? Before you panic, bear with me for 3 minutes. Let's start with sleep itself. We are wired to sleep at night, but how do our bodies know when night is? In our eyes, we have photoreceptors, cells which differentiate light from dark. Fair enough, but how would our liver or kidneys know the difference between day and night? Well, when it's dark, photoreceptors send the signal to the brain, to the pineal gland, which starts the production of melatonin. Melatonin is a hormone of the night. It's a chemical expression of darkness. It flows in our blood and lets all the organs know that it's dark and it's time to go to bed. Because of that, in many countries, melatonin is sold as a supplement for people suffering from insomnia. However, some time ago, a study was conducted where melatonin was fed to hamsters. Worryingly enough, as a result of melatonin intake, the hamster's testicles shrunk. The news hit the media, and panic ensued. You can imagine the headlines of tabloids. Men, if you take sleeping pills, your balls will be the size of peanuts. But will they really? Let's go back to the study. If we feed hamsters more and more melatonin, it's a sign for their bodies that periods of darkness, i.e. nights, are getting longer and longer. And what does it mean when nights are getting longer? Well, winter is coming. And in winter, hamsters, like many other mammals, undergo what is called daily torpor. Their metabolism slows down. Because it's cold and the food is scarce, they cut down all the non-essential parts of their body, such as testicles. After all, a hamster in winter is focused on survival and not impressing the ladies. So, yes, in winter, in hamsters, melatonin does cause testicles to shrink. But does that also hold true for humans? Well, no. We don't hibernate in winter. Men's testes are naturally the same size throughout the year. So I have 2 take-home messages for you. First, we need to be very cautious when interpreting the results of animal research because it's not always applicable to humans. And secondly, gentlemen, you can sleep peacefully. Your gonads shall remain safe.
Can there be bonus points for getting the word gonads in? I like that. So have there been any experiments done about people like living in permanent darkness? Like, you know,
Well,
Antarctic survey, you know.
So melatonin production is not only about controlling our sleeping patterns, it can also be linked with mood swings, and this is why people living close to the polar circles who naturally would be producing more melatonin sometimes are prone to depression more. On the other hand, we've got problems with people who are blind, who sometimes, depending on the damage to the eye, can't sense light. Sometimes they can because the connection between the eye and the brain is broken, but the photoreceptors are okay. So sometimes they would need to take either melatonin or other sleeping pills in order to regulate their body clock and function normally in the society.
So the obvious but tricky question is, why do we sleep then?
Why,
well, clearly it's not to make my balls smaller. But it's, there must be another reason.
Well, you've just held a big talk on it just before
I'm
this
primed
event.
For this. That's right.
Yes, yes. Well, what was it that Penny was talking about? To consolidate your memories. That was her favorite point. Yes, everyone, if you are learning something, if you sleep on it, it actually improves the results of either a task that you're doing or a poem that you're learning. It's actually fantastic to regenerate as well. Yes.
Do you think we might be making other mistakes by using animal models and not having that kind of reanalysis of the research that you've described?
I think it all depends on the research question that you have and on the message that you pass on to the public. So I am a conservation biologist, a behavioral ecologist. I work with animals. So of course I do animal research, but then my results are used to benefit other animals, and so they're very easily translatable. But if you do use animal models for medical research, of course, you can't make that leap straight away. And actually, scientists tend to be very careful when saying, we have discovered this and that. It could have implications for hamsters, and eventually might lead to development of new medicine for cancer in humans. But media tend to pick up on cancer, humans, hamsters, maybe, and bunching it all together in something that's completely obvious and completely definite, and that's when the trouble begins.
Blame the media.
Yes, I do blame the media, although a lot of the media are very wonderful and appropriate science communicators. So thank you, media, but be precise, please.
Yes, I'll take that personally. Time to say thank you and gonad to Joanna Bagniewska. I do think The Hamster in Winter sounds like a lost low-budget British movie as well. So is Joanna now in pole position for a place in the final? What about Adam, Ahmed, Anna, Caroline, David, Ding, Maria, Stella, Marius, Michael, Nikos, Porik, in alphabetical order? All were great, but only 41.66666% of them will be deemed sufficiently famelabulous to make it through to Thursday night's grand final. Doing the deeming is down to our judges. They need to be quick about it because we're supposed to be out of here in about 15 minutes' time, and people may have lives to go or basement windows to check for flooding. So judges, get thee gone. And if you want to send them on their way, last chance to shout out names, countries, gonads, whatever you wish to do. Thank you. Now, before we go into the plan bit, we have a little tiny bonus item. Joanna is going to come back, not to reassure men about something, but for something slightly more topical.
Oh, is my mic on? Yes. I just wanted to say that today, 4th of June, is marking the 25th anniversary of the first partially democratic election in Poland. So I'd like to wish all of you happy Freedom Day, happy democracy. Yay.
Now, it looks like the judges are still not quite back with us. So you get a little tiny bonus from me because somebody on the way in was saying, why is FameLab called FameLab? Surely scientists rarely get to be famous. Now, I could answer that question, but actually, there is a greater prize out there than fame. I'm just back from Japan, where I went to visit a small place out on Kyushu, the southernmost island of Japan. There's a city there called Kumamoto, and outside Kumamoto is a shrine. And every year, they have a festival there. Well, it's a small celebration to honor the person to whom the shrine is dedicated, who is known as the Mother of the Sea. And they put fruit and flowers and fish onto the shrine, and a Shinto priest performs an elaborate ceremony, and all the locals, which is a major fishing community, turn up and report how the year has gone. And if you're there and observe it as I did, you increasingly realize that they're not just honoring the person to whom the shrine is dedicated, they're reporting back to this person. And the Mother of the Sea is not some ancient Japanese deity or some water spirit. It's not even anybody Japanese. It's not even anybody who ever visited Japan. It's a British woman scientist who did her work in the 1940s. And she's a woman called Catherine Drew. And as I said, she never set foot in Japan. She worked on the North Welsh coast mostly, looking at algae. And particularly, she found out the breeding pattern of this particular kind of seaweed and discovered that what had been thought to be two separate species was actually one. And somebody Japanese, this is the nature of science, read the paper and thought, this sounds a bit like some of the seaweed we grow here, the edible seaweed we have here. And she found a way that she could, that by learning from this, that they were able to harvest the seaweed. This became the basis of the nori industry. The nori industry is the basis of the sushi industry. And thus, it became this billion-dollar industry. So this woman, Kathleen Drew, if you think fame is too much to hope for as a scientist, this woman is a goddess. That is your higher bar to go to. And with that thought, please welcome back our judges. So we've had joy, we have fun, we've had seasons in the sun and the rain, but now all good things must come to an end, including the debate from the judges. So Wendy, break it to us gently, but break it to us swiftly.
Right, okay, well, it sounds like a cliche, but it was incredibly hard. Everybody did a fantastic job of showing their passion for science, we thought, in incredibly creative and different ways. And they've all come a long way to be here. So, you know, we just think they've done a fantastic job just to be
Okay,
here.
round of applause while they're all united. Okay, we've had the skill, now move in for the kill.
Well, just before we do the last announcement, that I wanted to say that this is my 3 favourite things that I learnt tonight that I never knew before. I never knew that if someone's kidneys didn't function, you smelled of acetone. I think that's quite cool. That sleep can shrink your testicles. All going to remember that. And that one smile...
Yes, yes.
That's it, mine. Well, I've slept a lot. And that one smile is equivalent to 2,000 bars of chocolate. I'm going to remember that one. I don't know if the other judges want to comment on anything.
I love the moth, I love the singing, any singing and dancing I was completely swayed by. I love some of the gritty performances and the general sexiness that went on.
Yeah. Have you considered going to, say, see Chicago rather than coming to be a FameLab judge? Right, okay.
Next year.
I just loved, I thought the people who didn't use props set themselves a higher... Sorry. Yeah. I thought the people who didn't use props set themselves a tougher task in communicating, but some of the sort of poetic language that came out, the brain as an orchestra, I loved that. And I loved all the furry animals, although I don't think the furry liverworm is going to sell very much.
Okay, we've had the orchestra, but now we need the soloists.
Yeah, the marks were very close, so the top 5 are only a few points above the rest, but we did have to pick. So the people we've picked in no particular order to go forward to the final are, drum roll, Joanna from Poland.
Come on up. Come on up when you're called. Come stand here.
Okay, Caroline from the UK.
Keep edging closer. It's not a personal hygiene issue. It's just space.
Porik from Benelux.
Oh.
David from France. And finally, do we have one of those really dramatic long pauses? No. Finally, Dingley from Hong Kong.
Whoa. Oh. So that is it. Thank you. Brilliant job from Please Can We Thank not just Wendy, but our other judges, Martin Hood and Mark Lithgow. Thank you also to all the backstage people, to Duca, to Alison for the honking the horn and keeping the time as well. Let's clap, everybody. And thank you to yourselves for coming along on tonight, which will be remembered, of course, as the night of the Great Cheltenham Deluge. Congratulations to the fabulous FameLab 5 who've made the final. Commiserations to the 7 who didn't. Console yourself with knowing the judge was, as always, bizarre and arbitrary, and you can relax and enjoy the rest of the Science Festival. That should mean the lineup for our final is, if I've got this right in alphabetical order, Bogdan Georgiou, Caroline Shenton-Taylor, Ding Lee, Jennifer Fowley, Joanna Bagniewska, David Delila, Marco Farigo, Porrick Flood and Ricardo Mori. They will all be doing brand new presentations, not the same ones as they did at the semi-final. If you come back here tomorrow at 8:30 p.m. and you'll find out nothing because we're over in the EDF Energy Arena. So that's where to be for the grand final. Oh, and I forgot about Lyle Tomlinson. Lyle! You are on my piece of paper, Lyle. It's just I skipped over in my attempt to alphabetize. It's always a habit of mine. So EDF Energy Arena tomorrow, grand final. Hope to see you there. And one more semi-final, final time. Congratulations to all tonight's 12 national FameLabulous FameLab winners.